The endoplasmic reticulum, or ER, and the Golgi apparatus produce and process proteins. These proteins move from their site of production in the ER through the Golgi, after which they proceed to their destinations, either taking residence in the plasma membrane, or being secreted outside of the cell, or fusing with endosomes or lysosomes. Proteins from the ER travel in transport vesicles to the ER Golgi intermediate compartment, or ergic, and from there to the Golgi apparatus, fusing with a compartment called the cis-Golgi network. Distinct processing and sorting events occur in an ordered sequence within different Golgi compartments. However, the mechanism by which proteins move through the Golgi apparatus has been a long-standing area of controversy, with two distinct models proposed: the Cisternal maturational model and the stable Cisternal model. Coloring one of the compartments will help illustrate the Cisternal maturational model. In this model, vesicles fuse with the cis-Golgi network until it begins to mature and move into the Golgi stack, and then the ergic vesicles create a new compartment in its place. The compartments, with their proteins, mature to become the medial and then the trans compartments of the Golgi stack, where further protein modifications take place. The compartments of the Golgi gradually mature and progressively move through the Golgi in the cis to trans direction, carrying their contents with them. The trans-Golgi network acts as a sorting and distribution center, directing molecules to their final destinations. The proteins leave in vesicles, which bud off until this final Golgi compartment disappears and is replaced by a maturing compartment immediately behind it in the stack. The transport vesicles that are associated with the Golgi apparatus function to return Golgi resident proteins back to earlier Golgi compartments for reuse. In the other model, the stable Cisternal model, the cisternae themselves do not move through the stack. Instead, proteins are carried between Golgi cisternae in transport vesicles. The enzymes active in each Golgi cisterna are retained in that cisterna. Or, if the enzymes are transported out of the cisterna in which they function, they are recycled by a retrieval pathway. Both of these models explain some, but not all characteristics of Golgi transport, and further studies are needed to resolve the mechanism of Golgi trafficking.